Thank you very much for that kind introduction, Siri. And I hate to even be following that panel. It gives me a little bit of chest pain. When I read Bijal Trivedi's book, Breath from Salt, during the pandemic, she actually referred to Dorothy Anderson's paper. So I went back and read it. And that's when I created that slide with the red boxes, because it was probably like the 7 stages of grief that I went through reading that, knowing that we've known this the whole time and we're just now catching up. But so thank you very much to that panel for sharing their really painful stories. So we're going to switch gears, although it's also relevant to this talk, and talk about exploring nucleic acid-based therapy. And when I say nucleic acid-based therapy, that includes gene therapy and mRNA therapy and antisense oligonucleotides, which we'll talk about shortly. And for those of you who don't know, I am actually from Denver, and that is the view of downtown Denver from City Park, which I'm very fortunate to live 10 minutes walk from. So these are my disclosures, most of which are related to the fact that I'm the primary investigator on a number of studies, and I do a lot of clinical trial design consulting. And my other disclosure is this. So Siri mentioned it a little bit, but some of you are aware that on July 12th, I actually flew from Denver to Narita Airport in Japan and then on to Indonesia, where I first went to the forest where orangutans live to help talk to the docs there about looking for respiratory disease. And then I went to Medan to teach the docs there about respiratory disease and orangutans, which actually responds to CF therapies, and then taught at a conference in Yogyakarta, and then flew back here on Friday night. So if I'm a little sleepy and say a little crazy things, yeah, that was me last night, actually, so which is why I wasn't dancing. So our learning objectives for today are to characterize the barriers to the successful development of nucleic acid-based therapies, understand the current state of development of these therapies in CF, and then the unique challenges for potential research participants in these trials. So we'll go through and define the scope of the problem, including those most in need of these therapies, review the goal of therapies and the differences between the different approaches, and then discuss ongoing and soon to start trials. So if you think about where we've been in CF, for the first 70 years ago or so, we actually focused on treating the signs and symptoms of CF with drugs like Dornase alpha and inhaled tobramycin, so treating the thick mucus and the infection. Then in 2012, we actually moved to being able to treat the basic defect at the protein level. So the chloride channel is that protein that we were able to treat with CFTR modulators. But now, really, in order to treat those who can't tolerate, aren't eligible for those therapies, we're going to have to focus on nucleic acid-based therapies. And ultimately, to have a cure for CF, we're really going to need to do gene editing, as they've been able to do in sickle cell disease. So, of course, we're at the point now that at least in high-income countries, the majority of people with CF are eligible for variant-specific, so mutation-specific CFTR modulator therapy. And some of you will have seen this slide yesterday when Dr. Moss was presenting, and it's a little bit busy, but it's data from the CF patient registry. So on the y-axis is the number of individuals, on the x-axis is the year, and then the different colors represent the different CFTR modulators. And if you focus just on 2022, you can see that only 82% of people who are eligible are actually taking CFTR modulators. So there are people who qualify for these drugs but can't tolerate them because of things like severe rash or liver function test abnormalities or effects on their mental health. So even though they are eligible, they're not able to take them. Also, as Dr. Moss showed you yesterday, and you sort of heard referred to in the last presentation, CF is a disease that occurs in people all across the world, but the variants in people who are not of European descent have not been adequately studied, and therefore people weren't enrolled in the trials and didn't have early access to these drugs. And even now, because we haven't really recognized until recently that CF occurs everywhere, there are people who are still not diagnosed but probably have cystic fibrosis. And these different colors represent those numbers of people who are diagnosed in those various places across the world. So there are big gray spots where we haven't yet diagnosed people the way that we undoubtedly should be diagnosing them. But even those who are diagnosed, as Dr. Moss pointed out, a lot of them don't have access to the CFTR modulators, even if they were eligible, because of the cost. So we still have a lot of work to do, and we also need to find other therapies for those who aren't going to be eligible once we actually diagnose them. And it's critical to understand, and then those of you who are living with CF and aren't eligible already know this, but it's critical to see what difference that makes. So this is again data from the US CF patient registry. On the y-axis is the average number of pulmonary exacerbations, and on the x-axis is the year. So of course, in the United States, Alextezaiva, or ETI, Trikafta by trade name, was approved. And you can see what happened to pulmonary exacerbations in those people who could actually take ETI. So they dropped dramatically, almost 70%. But for those people who were not eligible for Alextezaiva, you can see that their exacerbations did drop during the pandemic because we weren't being exposed to as many viruses during that time, but they didn't drop the way that they did for those who were on Alextezaiva. And it's not just physical health that's impacted. So Emily's entourage, you've heard mentioned before, was started by Emily and her family to really focus on that group of people who weren't modulator eligible. And she did a survey and looked across the world at people's health experiences in the case when they weren't eligible or didn't have access to CFTR modulators. There's a lot of data in this paper, so I encourage you to read the whole thing. But what I want to point out here, again, is there isn't just a health impact, which is a big enough problem as it is, but there's also a serious mental health impact. So these are quotes from people who either have CF or their loved ones of people with CF who aren't able to benefit from modulators. So the first one is from a parent. The older my child becomes, the harder this disease becomes to manage, physically and emotionally. It's extremely hard to watch as it slowly takes over. Then from an adult with CF, I'm 42 and I feel like I'm running out of time. My only CF issue is my lungs. My last call with my doc, he said, I wish I had a modulator for you. Same, doc, same. And finally, from a partner of somebody with CF, I'm glad so many have access to CFTR modulators, but at the same time, I am concerned for my partner. I worry that 90% coverage could be seen as good enough, and I worry about the effect being passed over by these incredible drugs has on my partner's well-being, both physically and mentally. So we have these medical impacts, and we also have this medical trauma, which we'll hear about later, that exists for those who aren't eligible or can't take these drugs. And if we go back to looking just at the US, you heard yesterday that up to 94% of people with CF could ultimately be eligible for these therapies. But if you look at the group who isn't going to be eligible, it's predominated by people who have lower lung function, so they're sicker. They have lower income and are more likely to be on Medicare or Medicaid. They're likely not to be seen at a CF Therapeutics Development Network Center, therefore may not have access to participate in trials. And of course, because variants are understudied in people of color, they're more likely to identify as a person of color. So we really need to start looking again at nucleic acid-based therapies to address some of these issues. But as we think about enrolling people in clinical trials, it's important to think about what we've done in the past that's going to make that a bit harder. So, as you've seen previously, there's been historical exclusion of Black, Indigenous, and people of color in trials. And now we're going to be asking people who haven't been asked to be in trials before to participate in really difficult, time consuming trials that actually are often first in humans. So, they haven't even been tested yet in healthy controls. And that's an issue. Also, again, these are going to be oftentimes first in human therapies. So we have to really approach this with great care. We know for certain things like mRNA, which we'll talk about in a minute, redosing is going to be necessary, but antibody development could actually preclude redosing also for gene therapies. Also, early phase participation may prevent people from being able to be in the next study. It wouldn't prevent them from having the drug if it were approved, but it may exclude them from being in the study. Also, all of these approaches are currently being focused on inhaled therapies just to treat the airways, which of course is a huge issue for people with CF, but that means that people will still have CF in the rest of their bodies, unlike with modulators, which are an oral therapy that goes all over your body. So it's important when we're talking to people about these studies that we explain all of these issues and make sure that people are really informed in their consent. So, what is the real goal with nucleic acid based therapy? What we want to do is reprogram the cell to make functional CFTR protein by providing the correct instructions, whether that's with mRNA or DNA or an antisense oligonucleotide, which is just a few letters of that big alphabet you see that actually sort of covers up the mistake and allows the cell to make the correct protein. So those of you who watched the plenary in 2021, given by Hillary Valley, so Martin Mentz and Hillary Valley shared this slide. So they suggested that you think about CFTR as being a car. So the CAR is CFTR protein, and the different CAR problems are CFTR variants or mutations. So when you think about mRNA therapy, mRNA is very fragile. It doesn't last very long, but it could work if you give the correct message to the cell and then it makes the correct protein so it can go to the top of the cell and function as a chloride channel. So it's like having a rental car. You're going to have to give it over and over and over again indefinitely. With gene therapy, you are actually providing the cell with the correct copy of the CFTR gene. So it's all the instructions it needs to make that correct protein, that correct chloride channel. So that is like having a brand new car. However, depending on which cells that you get into, so there are different layers of respiratory cells, and the ones that we really need to get it into for it to last a long time are these ones at the bottom called the basal cells. So depending on how many of the basal cells we can actually get that corrected gene into, the longer it may actually last. But it's likely that we would have to redose gene therapy as well. And then finally, there's gene editing. Again, the example of that most recently has been sickle cell, where they actually take the blood cells out, edit the gene, fix it, and then give the cells back. We can't do that in CF because you can't take the lungs out and fix them and then give them back. But ultimately, that's what we really want to do in CF to cure CF. So that would be a permanent fix. So back in 1989, when the cystic fibrosis transmembrane conductance regulator, or CFTR gene, was discovered by a collaboration between Lapchi Choi, Jack Riordan, Francis Collins, and Batsheva Karam, amongst others, everyone thought, okay, we discovered the gene therapy will be around the corner. This is really straightforward. It's a single gene. We can fix this. Unfortunately, as you're well aware, that didn't happen. So what was the issue? Well, back in 1989, they discovered the gene, and almost immediately, people started to try to figure out whether we could do gene therapy. And there were actually mouse models that were used and showed that that actually possibly could work. At that point, we weren't really sure how many cells. They thought maybe 6 to 10% of cells could be corrected, and that would be enough. And they started using adenovirus as the vector, so the way to carry the corrected gene into the cells. But what they quickly realized is that adenovirus actually caused a lot of inflammation. And so not only could they not actually get enough of it into the cells, but also it was causing people to be very sick because it was causing too much inflammation. And so those trials really ended in 2001, and we realized that we really couldn't do it with that vector. So then people moved on to adeno-associated vector. So that's occurs naturally but doesn't cause disease in people. So there were 6 trials with AAV-based gene therapy in the upper and lower airways. So first they did it just in the nose, thinking if you can correct it in the nose and show that it's safe, then we could move on to the lungs. So they dosed about 34 participants between the 3 trials, and they actually did show that it was safe and well tolerated when just applied to the nose. And they showed that you could actually detect DNA. And there was an improvement based on nasal potential difference measure where we watched the chloride transport in the nasal epithelium. They showed that actually did improve. So they moved on to using AAV in the lungs. They dosed about 84 people who had mild to moderate lung disease. And they showed again that it was safe and well tolerated. We weren't seeing the huge immune responses that we're seeing with adenovirus vector. They again detected DNA, but transgene expression was not there, and there was no change in lung function. So again, unfortunately did not work. So while we were focusing on the CFTR modulators and their development, the UK team continued to work on gene therapy. So they actually decided to try putting the corrected gene in a liposome, basically a fatty ball, and seeing if that would actually work to get it into the cells in sufficient amounts to actually make a difference. So they did a phase 2 double-blind placebo-controlled trial of their drug, about 130 patients who were over the age of 12 and had, again, mild to moderate lung dysfunction. Everyone in the trial got monthly nebulized therapy for 12 months. They did actually show that there was stabilization in lung function. So the one in aqua is the group of people who got drug, and the one that's in red, again, sorry for those who are colorblind, but the other circles are people who got placebo. And one of the things that people noted about this trial is that those in the placebo group actually declined more than you would expect. So at that point, people were losing lung function on average at about 1 to 2% per year. So that was a pretty big drop. And so it looked like the drug was a little bit better than it was when you compared it to the placebo group, but essentially there was no change. So again, they looked at all these different outcome measures and saw no difference in any of them, although they didn't have a lot of difficulty with what we call treatment emergent events or side effects that we think are related to the drug. But at that point, everyone said, okay, we really don't have the technology yet to make this therapy work. We have to go back to the drawing board and think about it a little bit more. So when you think about preclinical, which means in cells and animal models, the question still remains is which vector is the right one to use? What do we need to get enough of the corrected gene or mRNA into the cells for it to work? So the different viral vectors we'll talk about in a minute. Also, which cells do we need to target? Do we just need to target the ciliated cells, or do we really need to get a lot of it into those basal cells that I mentioned, which are sort of the stem cells, the one that create all the other cells in the airway? And then what percentage of those? Is 6 to 10% enough, or is it 15%, or is it 50%? We don't know the answer to that yet. And then also, what efficacy, how much of it working do we need to see in cells and animal models before we say it's good enough to go into clinical trials? Because I told you that there are about, in the United States, about 6-7% of people who don't qualify for modulators, and then probably another 10% or so who don't qualify or don't tolerate modulators. So that's still a very small group of people in which we can test these different therapies. So we have to be very critical in our thinking about what's worth moving forward. In the past, as a member of the Clinical Research Executive Committee, we said, yes, bring all your trials to us. And now we're much more selective because we know we have a small group of people who are going to be eligible for these trials. And so we can't just move everything forward. We have to be very, very selective. So if we say there's enough preclinical data, what do we need to look for in early phase studies? So phase 1 studies or phase 2 studies. How do we measure efficacy? The standard in CF has always been percent predicted forced expiratory volume in 1 second, or FV1, so lung function. And the reason that we can use that is because a long time ago, Iten Karam and others showed that lung function is a surrogate for survival in people with CF. In other words, the higher your lung function, the longer you're predicted to live. And that's why the FDA will allow us to use FEV1 as a marker. But we know now that there may be some ceiling on how much you can improve FEV1. So is that the right one to use for these trials? Maybe. We don't know the answer to that yet. But what other things can we use? So some of you may have done multiple breath washout if you've participated in any trials. So that's basically seeing how long it takes a gas that they have you breathe in. How long does it take for that gas to diffuse back out? Well, that one hasn't yet been proven to predict survival. So the FDA has been willing to take that in little kids, but hasn't been willing to take that as an efficacy marker yet in adults. Also, we can't do sweat tests in the lungs, unfortunately. So we use that for the modulators, but we don't have a way to measure chloride transport in the lungs. There are some experimental things that are trying to be done, like measuring NPD, like in the lungs, so they call it LAPD, but that's not yet ready for prime time. Also, I mentioned that these vectors can often cause inflammation. So what measures of inflammation do we need to measure before we say, yeah, now we can go on to that bigger trial? And of course, what the FDA needs to approve drugs is phase 3 trials, the big trials where there's a placebo control. And again, what efficacy measures do we need? What is the FDA going to accept for us to get these drugs approved? And how much improvement is enough? So if you think back to the Kalydeco or ivacaftor trials, lung function improved about 10% on average in people who took ivacaftor. But if you looked at the Lumiva or Cambi or Teziva Symdeko trials, lung function only improved 2 to 4%. So is that enough? If this therapy costs, I don't know, the drug for sickle cell costs $1 million. So if it improves lung function 2 to 4% and it costs $1 million, is the FDA and our insurers going to pay for it? Or do we have to get levels of improvement that we saw with Teziva? Also, which people with CF are included in the trial? So do we only include people who aren't eligible for modulators? Or can we also include those who didn't tolerate modulators? And what about people who are on modulators but didn't have a big improvement in their lung function? These are all the things that we're thinking about as these trials are being designed. So let's move on now and talk about those drugs that are actually in clinical trials right now or soon to be in clinical trials. So there are a number of different mRNA studies. So Recode, Arcturus are ones that are getting ready to start. And the Vertex mRNA trial is already ongoing. And then also I mentioned antisense oligonucleotides, those little bits of information that sort of cover up the incorrect information and allow you to continue to make the correct protein. That one is getting ready to start. So I'll show you just a little bit about each one of these trials. But first I wanted to mention, and I'll do this with gene therapy as well, is safety. So all of these early trials are going to be first looking at, is this drug safe to move on to the next trial? And so we did relatively recently have an mRNA trial where they put the corrected mRNA into a lipid nanoparticle, or a little fatty ball basically, that people inhaled. And what we saw in that trial, so let me show you this part. Oh, it's not showing well here. Is all of the people who got the therapy versus those who got the placebo. And if you look at, again, the treatment emergent adverse events, there were mild symptoms, much more in the placebo group actually than in the treated group. But if you looked at the moderate symptoms, those occurred more in those who actually got the drug. And when you specifically looked at what kind of symptoms people were having, it included things like cough, headache, pyrexia or fever, chills, chest discomfort, pulmonary exacerbations, and wheezing. So the kind of things like you would think of having when you got an immune response to something. So it could have theoretically been a response to the mRNA, but more likely what people think is it was a response to that lipid nanoparticle, which is what you need to get it into the cells. So that's what we're looking for in these early phase studies. Are people having these type of side effects? So as I mentioned, there are 3 different mRNA trials. The first one is RECODE that I'll mention because that's the one that's not yet in trials, but it soon will be. So basically what RECODE's approach were to create these lipid particles that they put proteins on that made it more likely to get into the lung cells and be specific for the lung cells, because you don't want it just to go into every place in the lungs, because that's not where CFTR is supposed to be. And so we're trying to avoid putting CFTR where it isn't supposed to be and just get it where we want it. So they created this lipid nanoparticle that they call SORT that really just goes into the cell types that we want it to go into. They then treated human bronchial epithelial cells, in this case, human bronchial epithelial cells that were from a person who had copies of F508. And they actually showed, if on the y-axis is chloride transport essentially, and on the x-axis is increasing amounts of the drug. And you can see that with increasing amounts of the drug, there was more chloride transport. So they showed that the efficacy of their drug, at least in these cells, was about 75% of that was made with Trikafta, which is shown on the far right. They actually also looked at some ferret data. I didn't show you that in the interest of time, showing that mucociliary clearance actually did improve in the CF ferrets who got this drug. They completed their toxicology studies, and they are working on their phase 1 study in healthy volunteers, because the FDA does think it's okay to give mRNA to healthy volunteers. And they're expected to start dosing people with CF later this year. So the next mRNA trial I'll talk about is Arcturus. So they actually really focused on optimizing how their drug was going to work once it was in cells. Again, they're using a lipid nanoparticle to deliver this corrected mRNA. And it, like the others, will be aerosol delivery. So they also use the ferret model. I think most of you are probably familiar with the fact that CF mice don't actually get lung disease. You have to do other things to them to get lung disease. And so John Engelhardt at University of Iowa created CF ferrets, where he knocked out genes in some cases, but he also created ferrets who had the G551D mutation and ferrets who had the F508del mutation. So in this case, they were using the ferrets who had the G551D mutation. And if you look, because these ferrets get all of the things that people with CF get, so including really bad gastrointestinal disease, the way he makes sure that they stay alive is by giving them Kalydeco. So he used the example of a ferret that was off Kalydeco. There was also a negative control, so they labeled the lipid nanoparticle with a red dye, basically, so that, or with a label, so that when it actually worked, it would turn red. And then they had animals on Kalydeco, that's in blue, and then they had animals that got their drug, which are in green. And you can see that on the y-axis is percent lung clearance or mucociliary clearance, and on the x-axis is minute. So you can see that those who got the drug actually had a big increase in their mucociliary clearance compared to those who were just on Kalydeco or who weren't on Kalydeco or just got the carrier control. So they also are planning to start their study in CF later this year. They completed already the dosing with people without CF. And so when they say single ascending dose, that means they give a little dose and see how it works, and then a bigger dose and see if it's still safe, and then a bigger dose. And then there's also something called multiple ascending doses where the person gets multiple doses over days and they can get bigger and bigger doses over time. So again, they already completed the healthy control dose and now they're moving on to their phase 2 multiple ascending doses in people with CF. And so again, should be starting later in the year. So the next one is the VX-522 study. That's Vertex, and they've been working with Moderna. So you guys, again, all know Moderna, what Moderna did with mRNA for the pandemic. So they worked with Vertex to create another form of a corrected mRNA that will be in a lipid nanoparticle. And they actually already started that study in the UK, and it'll be starting, well, actually has started here as well, the multiple ascending doses. So first they tried it in healthy controls and showed that it was safe, and now moving on to people with CF. So the last one in this category of sort of mRNA and antisense oligonucleotides is Splicent. So this company was actually created by Dr. Karam, who I mentioned was one of the people who helped find the CF gene in the first place. So what they're doing is creating, again, these little messages, so it doesn't take nearly as much to get it into the cell because mRNA is big, not as big as DNA, but still big. So because this is so small, you don't actually have to use a lipid nanoparticle to get it into the cells. You can just give the drug. And so they're starting out with this specific mutation because it's a splicing mutation. And so the idea behind the ASO is to hide that incorrect splicing information so that the cell will keep making the protein. So in cells, they actually showed that you could get correction with this drug up to normal CFTR activity. That was in cells. They then checked in people without CF to make sure that it was safe, and it was very safe. So they could give 40 times the amount and it was still safe. And so they completed that phase 1, and they're now moving on to a phase 2 study in people with CF, again, with multiple ascending doses. So doses over days at higher and higher doses until they figure out what the right dose is. And then they'll move on, if it's successful in being safe and maybe showing a little bit of efficacy or working properly, they'll move on to a bigger study. And also, I should say that if they can show that it works in this particular variant, they may be able to show that it works similarly in other variants, but they're starting out with this one that they've tested and, again, shown in vitro, at least in cells, that it can correct all the way to normal. So let's move on to talking about gene therapy. So Spirovant and Crystal Biotech both have drugs that aren't yet in the CF therapeutics development pipeline. So Spirovant hasn't actually started their trial, and Crystal Bio they have their study at 2 sites, but it hasn't actually been approved by the CF Therapeutics Development Network. So I'll just say a little about those. And then also Boehringer Ingelheim has a drug that's actually going to be starting in the UK in its testing, and then 4DMT has been being conducted here in the United States. And so I'll spend some time on that one. Siri, how am I doing on time? I don't have a clock up here. Okay, so I should talk faster. Okay. Okay, so again, as I mentioned before, safety is always the first thing that we're checking on for these different trials when they're in their early phases. So some of you will have seen in the news and may remember that back when gene therapy was being tested in the early 90s and late 90s, there was a death of somebody who got AAV therapy, not CF, but got AAV therapy because of the huge immune response. It is important to recognize that these are really high systemic doses that were used in that trial, and we're using much, much lower doses and only in inhalation form at this point in CF. There was actually another recent death in somebody who had Duchenne muscular dystrophy, but again, getting very, very like 10 times as much of a dose that we're giving in CF. And then one of the studies I'll show you is using a lentiviral vector, and that one has been linked in the past to causing cancers over time because it actually does integrate, or at least with this study, integrated into the genome. So these are the kind of things that we have to think about. So when we compare these different viral vectors, there are pluses and minuses to each one of them. So with adeno-associated virus, or AAV, it has a much lower risk of pathogenicity, so it doesn't usually cause disease in people. It has a good duration of expression, so how long it stays in the cells. It can affect a broad range of organs, which is why they've tried it in other diseases as well. Generally, there's a lack of a strong immune response like we saw back with adenovirus, and there's a very, very low risk of it actually inserting into the genome, close to zero. The disadvantage is that AAV, the receptor for it, where it binds to the lung cells, is actually on the bottom and not on the top, so you have to get it all the way to the bottom of the cell. It has a really small carrying capacity, so you can't put the whole CFTR gene. You have to take little bits off to fit it into an AAV vector. There is a potential for pre-existing antibodies because it's here in the environment. There's a potential for hepatotoxicity at very high doses. Again, not so relevant for CF right now. And we're not sure that you can repeat dose it, because if you develop antibodies, then it may not work as well the second time around. With herpes simplex, which is what Crystal Bio is using, it isn't integrating, so that's good. It has also low chance of causing immune problems, and it has a very large carrying capacity, so you can fit the whole gene in there. And we know it's okay to repeat administer, but there's very, very little data, and HSV doesn't have a propensity naturally to bind to respiratory cells, so it may be more difficult to get it in the right place. And then finally, lentivirus, the most recent ones they've been making are inactivating, so we hope that they will not actually insert into the genome. They also, lentivirus doesn't naturally go into the lungs, so they had to do something called pseudotyping, which means putting proteins, adding them on to make it more likely to go into lung cells. It's low likelihood of causing immunogenicity. It does actually last a long time, and it has a large carrying capacity, so you can put the whole gene into it and you can repeat administer it. But again, there is that risk of inserting where you don't want it to insert and therefore causing cancers. So just running through these really quickly. So again, splice senses AAV. They actually have looked in ferrets, which is the next slide. And they've shown first in cells that it did improve in the setting of using doxorubicin. So in order to actually get it into the right place, they had to add this medicine called doxorubicin. And when they did that, it actually did work in cells. So chloride transport on the y-axis and increasing doses of the drug on the x-axis. And when they actually administered it to ferrets, they were actually able to get a large number all the way up to wild type or normal number of copies of the corrected mRNA or the DNA into the ferrets. So this study is another one where I told you it's not yet approved by the TDM. This is Crystal Bio's HSV vector, and they are actually doing it at 2 sites on the East Coast, looking at early safety. So BI is the study that I mentioned that's going on or going to start soon in the UK. So again, that's the lentivirus vector, where they put those extra proteins on the outside to help it get into the right cells. They actually did show in human bronchial epithelial cells that their drug does increase chloride transport in those cells. And they are now moving on to a study. This actually was given to non-human primates, so you can see the pink where they actually showed that they did get it into the right place in the airways. So in their study, they're actually going to do open label for this early phase. It'll be first in humans. So again, the FDA and EMA don't allow you to give gene therapy to healthy controls. They have to give it to people with CF. So it's a 24-week observation for the first part. And then they actually have to be followed out 15 years based on the FDA and EMA regulations, because they want to monitor people to make sure that they didn't have that insertional mutagenesis. In other words, insert into the genome and potentially cause cancer. So it's a very, very long follow-up. And then the last one I'll talk about is 4D710, which is AAV therapy. So this is the gene therapy that's furthest along. When you try to get things into the lung cells, obviously in people with CF, there's lots of mucus and there are antibodies. And so they designed this specifically to get around all of these problems. So this A101 capsid, the way they actually get it into the cells, was tested in multiple non-human primates as well, and showed that it actually did get through the normal mucus, at least in these non-human primates. But they did have to take off, as I mentioned before, little parts of the CFTR gene because it wouldn't all fit in. So it's currently in still early dosing. So we started out at a dose of 1 × 10⁻¹5 vector genomes and gave that to 3 people. We tried to go a little bit higher, as I'll show you in the next slide. And then we also cut the dose in half and then cut it in quarter, because what you want to do is use the lowest dose that produces safety and efficacy. So we're trying to figure out what that dose is. So per the FDA, folks are being followed out for 24 months. So this is sort of a busy slide, but it shows people who got 2 × 10⁻15 vector genome dose, the highest dose, 1 × 10⁻15 vector genomes, and the lower doses. And because the lower doses have just started being tested, we don't have as many people that are in those. On the bottom part of the slide, I'm showing you controls, so non-CF lung and CF lung. That's just commercially available CF lung. So at that very highest dose, we did see 2 people who had adverse events in retrospect, and we think that's because we got too much CFTR in there. So what I'm showing you with those arrows is that it actually got in the interstitium. So basically not in the airway cells, but in the part in between. And that wasn't where we wanted it to be. And it may be just because it was so effective at that really high dose that we thought that dose was too high. Although some people actually did well on that higher dose, because 2 people didn't do as well, we decided not to move that forward. So all the people who got the next lower dose, the 1 × 10⁻15, tolerated the drug just fine. And we did actually show the brown staining is where the CFTR protein is. And so far, the participants who got the next 2 lower doses also had correct staining in the right place in the airway cells. We also looked at lung function out to 12 months, and we only have that for the first 2 cohorts because the other 2 aren't out to 12 months yet. And all the little circles indicate that somebody got a respiratory virus right before or within 28 days of their lung function. So you can see that in those people, even though they got some viral infections, because this was right after the pandemic that some people got diagnosed when everybody was getting respiratory infections, those people with the lower lung function actually did have an improvement that was sustained out to 12 months. And generally, the people who had higher lung function to start stayed stable. The other thing that we looked at, because the FDA cares about how people feel, function, and survive. So survival is FEV1, and function is things like how much exercise you can get, and how you feel is measured by the CF Quality of Life Respiratory Questionnaire. So we know that the minimal clinically important difference, like the smallest amount that you can improve that really means anything, is 4. And you can see that whether you looked at the individual data or you looked at the data that was combined, generally people had improvements in their quality of life, and it was above that minimally clinically important difference. So for those of you who haven't seen this before, it's the CFTDN development pipeline. So All the phases of studies are on the y-axis, and then the different kinds of studies are on the x-axis. And you can see that there's still a ton that is being done. I think I have 3 more slides. So I'll hurry past those. Okay, this one I just wanted to show really quickly. So gene editing is still very much in the preclinical phase. So this study that was just published in Science earlier this year showed that they could give this gene editing drug systemically, so through the whole body, to these mice. And if you look at the red again, these mice had a dye that turns red if you actually get the gene to the place that you want it to in the lungs. So you can see all the way out to 660 days, those mice who got the systemic gene correction or gene editing still had the correct gene out to 660 days. So that's exciting. But again, this is still in mice. So we still have work to do, because obviously if you're editing genes, you want to absolutely make sure that you're only editing the ones that you mean to edit. So continue to watch this data. I just wanted to end by going back to Emily's study. So the whole reason that we're doing all of this is to help people with CF, but we do need people with CF to participate in trials. And so Emily asked, for those people who aren't benefiting from modulators, what are their motivations for participating in trials? And so the top one was the possibility of improving health. But I want to point out that the next 2 were the desire to help find a cure and the possibility of helping the CF community. So if that doesn't give you hope, I don't know what does. And I think that both Skylar and Trey talked about this. So Skylar gave permission to show this. But when I asked him about why he participates in trials, he said it provides hope. The research science makes sense. Waiting on the results is worth it. And you're making such an important impact on the future of those affected by CF. And then Trey also said something similar. So participating in research, I feel like it's building blocks to help the CF community. I feel like it gives us hope. So with that, I'll conclude saying that highly effective treatment is available for a lot of people, but certainly not for everybody, and it's not a cure. We've learned a lot of lessons from previous gene therapy work, and we're applying those to what we're doing now. As investigators, we have to provide really clear information to people who are thinking about participating, and you all need to ask the tough questions of the people who are asking you to participate. A number of different approaches are being developed, and these ongoing and upcoming trials are really going to teach us a lot about our ability to correct the gene and really cure cystic fibrosis. So thanks to all the people with CF and their families who have bravely participated in these trials, the research centers, those people who donated slides, my team who has been holding down the fort for the last 2 weeks while I have been out of the country. And yeah, I'll take questions.